Good afternoon to everyone joining us from Spain, the European Union, and the international community. I am Victor Santana, a collaborator of the Fundación Longenia in the area of communication. On behalf of Fundación Longenia and the Bilu Huang Institute for Aging Research, it is a pleasure to welcome you to the inaugural session of our joint scientific webinar series. This series marks a milestone in our strategic international partnership built to advance translational longevity science and foster rigorous open debate based on first principles. Fundación Longenia works at the intersection of cutting-edge science and technology to mitigate biological decline and expand human anatomy autonomy. Our partner, Bilu Huang Institute for Aging Research, based in Hong Kong, is an independent nonprofit research institution dedicated to theoretical modeling and inquiry into cellular senescence. I'm not sure if that's how you pronounce that word, but senescence. Yeah, senescence. Cellular senescence.
Yes.
Today, we are privileged to introduce the leadership team behind Bilu Huang Institute for Aging Research. Bilu Huang is an independent theoretical researcher in aging biology and the founder and principal investigator of the Bilu Huang Institute for Aging Research. He's the originator of telomere DNA and ribosomal DNA co-regulation model for cell senescence, a theoretical framework for understanding aging and cellular senescence. He also serves as an honorary scientific advisor to Fundación Longenia. And then we have Claire, who is the executive director and co-founder of the Bilu Huang Institute for Aging Research. She holds a master's degree in economics from Waseda University, Japan, and has professional experience in the pharmaceutical industry. She works at the intersection of scientific communication, international collaboration, and nonprofit research management, supporting the development and dissemination of aging and longevity science. Then we have the scientific core of this episode is titled, Why Do We Age? The TRCS Framework for Cell Senescence. Moving away from traditional views that define aging as a random accumulation of damage, the TRCS model proposes that cell senescence is an intrinsic genetic program. It is driven by the programmed shortening of telomeres and rDNA arrays via the p53 pathway. This unifying theory offers a clear framework for experimental investigation and highly testable predictions. Following a 30-minute scientific presentation, we'll host a 20-minute joint Q&A session. Without further ado, Claire, Bilu, the floor is yours. Thank you very much.
Thank you, everyone. Hi, everyone. And good evening, everyone. On behalf of BHIAR, I would like to thank Fundación Longenia and Mr. Cayetano Santana Gil and Victor Santana for their support, coordination, and hospitality in organizing this webinar series. We are delighted to have this opportunity to share and discuss the TRCS framework with colleagues and friends interested in aging biology and longevity science. Seated beside me is Mr. Bilu Huang.
你好，我是毕路黄。
The originator of the TRCS framework and the founder of BHIAR. I will be presenting the webinar in English on his behalf. During the discussion and Q&A section, I will also serve as his interpreter to facilitate communication with our international audience. Thank you all for joining us today. Yes, so disclosure. Before we begin, I would like to provide a brief disclosure statement. Bilu Huang serves as founder and chief scientific officer of Fuzhong Therapeutics. He also serves as honorary scientific advisor to Fundación Longenia in Spain. I serve as Chief Operating Officer of Fuzhong Therapeutics. The views presented in this webinar represent our current scientific understanding and are intended solely for academic discussion and open scientific evaluation. Next, please. Yes. Before discussing the TRCS framework, I would like to briefly introduce BHIAR. The Bilu Huang Institute for Aging Research, or BHIAR, is an independent nonprofit research institute incorporated in Hong Kong SAR, China. The institute is dedicated to theoretical research in aging biology, age-related diseases, and translational longevity science. Our work focuses on first principles inquiry, scientific communication, and international collaboration. BHIAR is also the originating institution of the TRCS framework that will be discussed today. Our mission is simple: conquer aging via first principles science. Before moving to the scientific content, I would like to briefly explain the background of the TRCS framework. The framework presented today is the result of more than 3 decades of theoretical research by Mr. Bilu Huang on the fundamental mechanisms of aging. As early as 1998, he proposed that the shortening of tandemly repeated DNA arrays, such as telomeres, may contribute to organismal aging. In 2011, he systematically proposed the mechanisms by which telomeres drive the progression of growth, development, and aging. In 2021, these ideas evolved into the TRCS framework, which proposed that both telomere DNA and ribosomal DNA may function as biological countdown substances in cellular senescence. More recently, the publication of Causality of Aging Hallmarks further developed the framework and its interpretation of aging biology. With this background in mind, let us now turn to the central question of today's presentation: why the problem of aging remains unsolved to this day. There are hundreds of theories attempting to explain why we age. However, neither any of these theories nor the corresponding interventions derived from them have been proven to significantly extend the lifespan of wild-type mice. Next, please. Limitations of current aging theories. Many existing aging theories seem plausible but fail to stand up to experimental verification. For instance, the free radical theory of aging proposes that reactive oxygen species attack biological macromolecules and render them inactive, ultimately triggering aging. The metabolic waste accumulation theory holds that the buildup of hard-to-degrade cellular debris, such as lipofuscin, beyond a certain threshold, leads to senescence in cells and organisms. The theory of accumulated mutations in mitochondrial and nuclear DNA states that excessive accumulation of mutated DNA drives aging. Nevertheless, antioxidants, which can scavenge free radicals, reduce lipofuscin levels, and lower the burden of DNA mutations, have failed to substantially prolong the lifespan of mice. Alpha-lipoic acid, a type of antioxidant, was even found to greatly shorten the median lifespan of mice. The epigenetic theory of aging argues that the loss and dysregulation of epigenetic information cause aging, yet partial epigenetic reprogramming has not succeeded in extending the lifespan of wild-type mice. Next, please. Tissue cells in multicellular organisms fall into two major categories: terminally differentiated cells and adult stem cells, which account for a small proportion of total cells. Both types of cells can be eliminated by the immune system due to cellular senescence, gene mutations, viral infections, and other factors. Adult stem cells then replenish lost cells through self-replication and differentiation. However, adult stem cells have a limited number of replication cycles. With each division, daughter cells are slightly older than their parent cells, a process defined as replicative senescence. Ultimately, the replicative senescence of both adult stem cells is the root cause of organismal aging. So, what triggers cellular senescence in the first place? Aging theories are generally divided into two camps: damage accumulation theories, including entropy increase and wear and tear, and programmed aging theories. Evidence from the life cycle of organisms and the replicative senescence of cells collectively indicates that aging is essentially a programmed process. If aging is governed by a biological program, a biological countdown substance must exist to drive this program. The replicative senescence theory suggests that telomeres act as a biological timer limiting cellular division. Even so, there are cases where telomeres do not shorten in certain species or specific cell types within the same species, while cells still have a finite replicative capacity. Moreover, maintaining long telomeres via telomerase cannot prevent cells from ceasing division eventually. This implies that besides telomeres, another biological countdown substance may exist in the cell nucleus. Against this backdrop, Bilu Huang proposed the telomere DNA and ribosomal DNA co-regulation model for cell senescence, the TRCS. In the TRCS model, multi-copy tandemly repeated telomere DNA and ribosomal DNA serve as the core countdown substances, like the sand in an hourglass. The model posits that progressive shortening of telomere arrays and/or RDNA arrays leads to a progressive increase in the level of the tumor suppressor protein p53. Since p53 binds to the promoter and enhancer of numerous genes, it continuously downregulates the synthesis rate of ATP and proteins over time. Meanwhile, it triggers specific upregulation or downregulation of various genes. For example, 1,500 genes show altered expression in senescent hematopoietic stem cells. This cascade drives programmed gene expression and gradually drives cells from a youthful state into a senescent state. Interpretation of aging hallmarks by the TRCS. In the TRCS framework, the shortening of telomere DNA and rDNA arrays acts as the upstream initiator of cellular senescence. p53 functions as the intermediate mediator, and all characteristic manifestations of cell aging are downstream events regulated by the countdown substances through p53 pathway. Next, please. The testable predictions of the TRCS. Core prediction 1. The p53 level is inversely correlated with the length of telomere arrays and rDNA arrays. Shorter telomeres and/or rDNA arrays correspond to a higher p53 concentrations. Prediction 2. Replenishment of telomere DNA and/or rDNA can reverse senescent cellular phenotypes and restore cellular replicative capacity. Prediction 3. Cells with longer telomere arrays and/or rDNA arrays exhibit a youthful gene expression profile. Gene expression patterns change progressively as telomere and/or rDNA arrays shorten. Next, please. Current limitation of TRCS. Comprehensive validation of the TRCS model is still lacking. Further research is required in the following aspects. 1. Monitoring changes in rDNA copy numbers during cellular senescence across diverse species, including plants, nematodes, and humans. Exploring the reconstruction of telomere and rDNA in germ cells after meiosis in these organisms. 2. Conducting large-scale longitudinal measurements and single-cell resolution analysis of the rDNA shortening in human cells. 3. Elucidating the interaction between telomere shortening and rDNA compensation, as well as the mechanisms underlying telomere and rDNA replenishment in germ cells. 4. Performing comprehensive rescue experiments to verify how replenishing telomeres and rDNA arrays affect aging biomarkers and lifespan at the cellular and organismal level. We hope the scientists around the world will help test and evaluate the TRCS framework. Through open scientific evaluation and experimental verification, we may move closer to understanding the fundamental mechanisms of aging and ultimately contribute to the realization of healthy longevity for humanity. Next, please. Thank you very much for your attention. We will now move on to the Q&A and the discussion section. Thank you. Are you muted, Cayetano?
Thank you very much. Any question? Nice to meet you, Michael.
Oh, Dr. was muted.
There it is. My pleasure, Gaetano. We've been in contact before on LinkedIn. It's nice to see you again. Thank you. My main question is the same one that Bilu and Claire have and you have, which is, when can we take this to actual testing in human patients? I think we're on the verge of doing exactly that, both through BHIAR and through what we're doing and what you'd like to do. I think it's exciting times ahead, and we'll just have to see how it comes out. The next question is, when will Bilu and Claire and I, all of our groups, be able to take this to human testing?
That's what we are waiting for, the real human clinical trials for telomerase. Bilu, do you have any comments? 那个 Dr. Fossil 说，大家都在期待等这个端粒酶疗法什么时候可以进入人体临床试验？就为什么这么大家等那么久？你觉得？
这个端粒酶，首先，是因为大家认为会致癌，还有就是因为小分子端粒酶几何基因对小组寿命没有延长。
So Bilu says that, well, the major question is that many people think the telomerase therapy may lead to cancer, and the second question is because of the small molecule telomere activator does not work well on extending lifespan of the mice, so that make people hesitate and doubt the telomere therapy.
I think that Bilu and Claire and I suspect that the risk of telomerase is overstated. The risk for cancer is overstated.
Yes.
Because it may be that by directly addressing a ribosomal DNA and addressing telomerase, we can accelerate DNA repair and therefore minimize the risk of mutagenesis and cancer. But still, all of that remains to be seen. And again, I think what we need to do is go to human testing. Still, I think that there's much to be gained by going after telomerase and ribosomal DNA, and that the risk of cancer is far overstated. The data actually doesn't support it, doesn't support that risk.
其实端粒酶，你说那个你看到 Fossil 教授写的那个吗？他是说，Fossil 教授认为端粒酶会致癌这个风险被过度
夸大。
而且其实相反的是
其实是相反的。端粒缩短才会致癌。
Yes. Also, I have interpreted for Bilu, and Bilu also says that the risk is exaggerated. And he also believes that it does not lead to cancer, and conversely, the telomerase therapy will not lead to cancer, and actually the shortening of the telomere will lead to cancer. So it's the opposite.
And I know Bilu and I have agreed that it's not a matter of theory and suspicion. The question is the data.
Yes.
As I did 30 years ago at National Institutes of Health, anyone who thinks you can reverse aging or thinks you can't reverse aging is naive. The question is, what does the data show? So I know Claire and Bilu and I want to see data.
Yes, because I remember it's also from Spain that Professor Maria Blasco's lab, it has used AAV mediated with their therapy.
AV9 and mTERT, right?
Yes, it did not cause cancer, and actually, reduce the incidence of cancer.
Yes, Maria and I agree on that, and so does Bilu. So do you, Claire. We'll see.
Yes, but I think that maybe because of the competition, and also say the speed, due to the fast speed of Chinese clinical trials, and we have seen some policies making the FDA is changing it, is also speeding up the clinical trials in the United States. So we really hope that the funding can come to this field first and accelerate.
Espero que sí.
Yes.
I hope so too.
And we also, do we have questions from Walter and Scout?
I think you've said things too well. No one has questions because you've done such a good job.
Thank you. I've practiced because it's not my mother tongue. So.
日本語がわかりますか？你会说普通話吗？
Oh, your pronunciation is very good. I know Dr. Fosso can speak nine languages.
Not very well.
Actually, Bilu has stated this question in his recent work on the TRCS framework, core mechanisms, testable predictions, common questions, and current limitations. And yes, does telomerase therapy induce cancer is really a frequently asked questions. So here's Bilu's. I want to share Bilu's. I want to share Bilu's further and systematic analysis to this question. That the prevailing theory holds that telomere shortening restricts the proliferation of cells carrying cancer mutations, thereby exerting a tumor suppressive effect. This has led to a common concern that anti-aging interventions relying on telomerase to extend telomeres may initiate or promote cancer, which greatly hinders the application of the telomerase-based anti-aging strategies. In fact, telomerase is expressed in all adult stem cells within tissues. Hence, although telomerase activity is a hallmark of cancer, it is not a driver of tumor. The following evidence demonstrates that the telomerase does not induce or promote cancer. 1. The immortality of cancer cells is independent of telomere length. Instead, it is associated with the TP53 mutations or excessive degradation of P53, even when the P53 gene remains intact. 2. Many tumor cells have shorter telomeres than healthy senescent cells. For example, telomere lengths range from 8 to 13 kb in normal ovarian cells, while it is less than 8 kb in ovarian tumors. The mean telomere length is 7.8 kb in normal hepatocytes and 5.2 kb in the cancer cells. And 3, telomerase activities is higher in younger individuals than the elderly, yet the incidence of cancer is much lower among the young. 4, a single tail vein injection of the AAV9 encoding mouse telomerase reverse transcripts in mice, reduced DNA damage, improved tolerance, and delayed tumor onset, and increased median lifespan by 24%. 5. Mice with ultra-long telomeres exhibit less DNA damage, a lower tumor incidence, and a longer lifespan. And there are also many other evidence, along with the evidence presented by Dr. Fossel, that actually extending telomeres can prevent cancer. I think maybe there's also another question that make people doubt the effect of telomerase therapy for anti-aging, and also the corresponding telomerase aging theory is that people use mice, this model, but mice have initially very long telomeres, so that is not very effective. If we move to other animal models or humans which have shorter telomeres, we believe maybe the telomerase therapy for anti-aging can do much better.
Incidentally, Bilu and I have a book coming out next year out of Singapore from World Scientific, and Bilu is writing the first chapter on how aging works, why aging works. In fact, Bilu, I just sent that off to the publishers. Now they'll get back to you with all the revisions, but it'll be coming out next year. Thanks for having Bilu on it.
就是麦克说你们明年那个书会出版，你要谢谢呀。你有什么想说的吗？对麦克，就我会帮你翻译。
没有什么想说的。
你可以表达一下感谢。
谢谢麦克。
Bilu just yes.
谢谢你。
So I'm wondering because My question, maybe for Cayetano or Dr. Foto, because maybe you are well-known, we are well connected with Dr. Maria Blasco, and I also know that she has founded a biotech based on the telomerase therapy in Spain. So how is that going? And I know the indication is also age-related diseases, maybe in cancer or in lung or liver.
Yes, I don't know. I haven't kept in touch with Maria for the past couple of months. I know she's been working on idiopathic pulmonary fibrosis. That was the initial target as she moved away from mice toward humans. And I don't know how it's going right now, frankly. I'd love to know.
由于小鼠端粒很长，
嗯
在小鼠上试验效果不如人，因为人端粒更短，端粒位的话应该会更好。
As Bilu just said, what I just said, that because the mice has initially very long telomeres, so we believe the effect for telomerase therapy will do much better on humans, as humans have much shorter telomeres.
Yes, mice, some varieties of mice have telomeres 10 times longer than mine at birth, while I have a lifespan on the average 40 times longer than theirs. But there's a good correlation between the speed of shortening of telomeres and lifespan of a species. The telomere length doesn't correlate with species lifespan, but the rate of shortening does. There was another article Maria put out with her group on this a couple of years ago. Good article.
Exactly. Yes. But I have...
So it's obviously more complicated than just length. There's more to it. Sort of thing that Bilu talks about.
Yeah, so the shortening rate rather than the initial length.
That was her argument. And it's obviously that's closer to the truth than just length. It's not a static measure. It's how fast you lose it. But again, it's more complicated. We'll have to see.
I have seen a paper say that there is research for the genetically engineered mice with telomeres, the length is genetically engineered to the length of the human. So maybe that makes that kind of mice model more adaptive.
Yeah, I know we have that same issue with regard to Alzheimer's research, where people will try to take a human beta amyloid, for example, ApoE2, ApoE3, ApoE4, try to take a human gene and put it in the mice, and you find that you don't get reliable results. And I think the problem is that, a human gene doesn't function by itself. It functions in the context of hundreds, thousands of other human-related genes and processes. And I think the same thing's probably true of telomeres. It's not simply a matter of M-tert and H-tert or telomere length, and it operates in a very complex cascade of processes. So yes, there's a lot to learn. I'm sorry, I'm talking too much. I'll be quiet.
你有什么想跟大家讨论的吗？
没什么可讨论的，可以在论文里看嘛。因为讨论毕竟还是说不清楚的。
It's like 你有什么问题或者什么都可以。
没有。因为该说的都写在里面。
Bilu says that he is more good at elucidating in writing, so he says he thinks that writing or rebuttal is more clear. Rebuttal or expressing is more clear in writing because it can make the corresponding evidence. So
agreed.
So
Cayetano. Es todo, tiene otros problemas, otras discusiones.
Any question more? No?
No más preguntas para mí.
Well, Thank you very much for the presentation of the TRCS model. We hope to have more participation in the second session. Thank you, Michael.
Zaijian, Shesheni. Thank you.
Thank you, Dr. Fossil. And gracias, Caetano and Victor and Scott and Walter.
Zaijian, Shesheni.
Thank
Gracias
you.
por todo.
Thanks.
See you soon.
谢谢大家，谢谢，
好
拜拜。